Welcome to Eye on the Cure, a podcast from the Foundation Fighting Blindness, providing science research, news, and insights about the world of vision and blinding diseases.
Welcome, everyone, to the Eye on the Cure podcast. I'm your host, Ben Shaberman with the Foundation Fighting Blindness, and I'd like to thank Boehringer Ingelheim for sponsoring this episode. And for this episode, I'm delighted to have a conversation with Dr. Deniz Dalkara. She's a research director at INSERM in France and leads a team in the development of gene therapies and models for neurodegenerative diseases at the Vision Institute in Paris. And excitingly, she developed a gene-agnostic gene therapy to resurrect visual function in dormant cones for people with retinitis pigmentosa and potentially related conditions. And for listeners, remember, cones give us central vision. They give us the ability to read and perceive details, and they also give us color. So resurrecting cones could be a really big deal. And what's exciting about her work is that the company SparingVision, also in France, has moved the treatment, it's known as SPVN-20, into a clinical trial at a few sites in Europe, and they plan to add sites in the US next year. And just an FYI, as many of you may know, SparingVision also previously launched a clinical trial for a different therapy, SPVN-06. That's preserving cone function, or at least that's what it's intended to do. So we'll be comparing Deniz's approach for resurrecting dormant cones and the other approach for preserving cones later in our chat. But first, Deniz, it's great to have you on the podcast.
I'm glad to be here, Ben. Thank you for having me.
It is my pleasure, and I think our listeners are going to enjoy what you have to say. And I want to give a little more background on Deniz before we start our conversation. She was awarded the BioValley PhD Thesis Award for a method of protein delivery developed during her graduate studies. And that was at the University of Strasbourg in France. And later, she conducted a postdoctoral fellowship in the lab of Ernst Bamberg at the Max Planck Institute of Biophysics before moving on to UC Berkeley to do a second postdoctoral training in 2007. And at UC Berkeley, she applied viral engineering principles to enhance AAV vectors for their application in retinal gene therapies. Then after that, she received a Young Investigator Award to start her work in Paris in 2012. Deniz also went on to receive a Euretina Science and Medicine Innovation Award in 2013, and she was also selected as a top innovator under 35 in France by MIT Technology Review in 2014. So Deniz, going through your background and the work you've been doing with SPVN-20, you clearly enjoy science. You're kind of a scientist. What got you into science? At what point in life did you say, hey, this is what I want to do?
So Ben, it turns out my parents are from a scientific background. So my dad is an MD PhD, and he's been very enthusiastic about research and science. So I think I've been exposed through my early life to a lot more science than humanities by way of upbringing. But then later it was this course in high school, an advanced placement biology class, that really got me interested deeply in molecules and biology and how all of that operates to make life happen. So I guess those are my earliest recollections of how I got interested in the field that I work in currently.
Certainly. Yeah. Having parents who are into science or scientists themselves certainly can be an influence. So I want to fast forward through your education to your PhD work, because that really started informing your gene therapy development. And can you tell us a little bit about what you were doing for your PhD?
Sure. So I first did a master's in pharmacology and pharmacochemistry, and then the way was open towards some possibilities. And it turned out that this was early 2000s and genetics was a thing, and everybody thought doing something related to genes was an intelligible path to pursue. And it turns out there was this laboratory working on delivery of genes into mammalian cells, right where I did my master's, and I wanted to continue in that laboratory towards pursuing a PhD. And one of the new things that was being thought of in that lab was now we've been delivering in vitro nucleic acids to cells. Could we also try to deliver directly the end product, so proteins, into cells using some of the same chemical methods that they'd been developing for gene delivery? So that's where I came in, and that was what was offered to me as a PhD study, and I enthusiastically pursued in that direction. Actually, the approach I developed got later patented and marketed by the spin-off of my PI's at the time, Jean-Paul Baer, and that was called Polyplus transfections, and it became this protein transfection reagent called Pulse-In, which found utility in a lot of little biotech applications.
That's really cool. And can you tell us how that protein delivery method is being used, for what conditions and what targets in the body?
So, like I said, these were relatively large packages of protein that we were delivering to cells in cultures. So this was more for investigational purposes that the Pulse-In product was being marketed. So now, more lately, there's been thoughts about using this kind of approach to deliver CRISPR-Cas9 into various cells in the body, but that hasn't quite gotten to the level of clinical application that we would envision using it quite now. But I would see definitely some applications coming in that area.
That's great. That's great. So it's boosting research at this point, but it sounds like there could be human application. So when you finished your PhD doing this innovative protein delivery work, did you know right away you wanted to get into gene therapy development? Because gene therapy at that point in time was still pretty cutting edge.
It was. I actually didn't even imagine that it would come this far back in the days, but I was a little frustrated with just working in cell cultures, and I was hoping to get access to more in vivo applications of gene delivery and gene therapy. And I guess that's kind of what brought me towards my next steps.
Got it. And so we're obviously going to talk a lot about gene therapy and its different forms moving forward. And I thought it might be helpful if you could give our listeners a simple definition of gene therapy.
So gene therapy in a nutshell is the utilization of nucleic acids, genes, for a therapeutic purpose. So in its most conventional form, the first thought of what comes to mind when we say gene therapy is gene replacement or gene supplementation. So this is the most straightforward approach. We have a mutation in a gene and it's causing the deficiency of a certain protein that is essential in a cell. We put back a healthy copy of that defective gene and it will make the protein and put everything back in order in the cells in which it's expressed. So that's what we think of most frequently, but really we can broaden that approach to think of many other applications. So this could be editing genes now that CRISPR-Cas9 and gene editors are available to us. So going right into the chromosomes and excising mutations right in situ at their source. This could be a gene therapy, or it could be just gene-agnostic therapy like the ones we're developing in SparingVision. So secretion of trophic factors to help cells thrive, survive across the disease stages for which we don't really have any other solutions like gene replacement, or we intervene a little bit further down the line. Really more symptomatic treatments can also be envisioned using genes as a therapeutic modality. So all of that can be, I guess, collected under this big umbrella that we call gene therapy. It's really using nucleic acids for therapeutic purpose.
Right. And it seems like for all these applications, viral delivery seems to make the most sense, at least at this point in time with the technologies. we have. And why are folks like you, scientists like you, using these engineered viruses?
So we're using what works already in nature. So we've been thinking about gene therapy as humanity for, several decades, certainly, but we haven't been thinking about it as long as viruses have been thinking about it. Viruses have had billions of years of evolutionary advantage over our rational thinking, so they're clearly much more clever at delivering or smuggling their genetic material into our cells than us. So I think we have everything to learn from them, and that's why viral vectors have been so useful for learning how to deliver genes. But of course, they also come sometimes with a problem that, we co-evolved with these viruses, and usually they're pathogenic, and our body has worked at ways to not let them do what they want to do, which is multiply endless copies of themselves. So we have to really be careful when we work with viruses for therapeutic gene delivery. We have to make sure that we get rid of all of the properties that are potentially harmful to humans and use them just as vehicles that deliver what we want.
Right. And that's been part of your work. I don't want to get too far ahead of ourselves, but making these gene therapies work better and more safely is kind of what you've been
so that's what the engineering is about. Yes, that's right. So if we use what these so-called garden variety viruses that exist out in nature, we're obviously using something that is suboptimal for our purpose. So the virus that we're using most frequently in gene therapy in the retina, as you know, is adeno-associated virus. And this virus hasn't really evolved to infect retinas. So we're really asking it to do something it usually doesn't do and hasn't really specialized to do. So in order to help the virus do that better, we can apply some bioengineering principles. We can even have it go through a little bit of evolution in the laboratory so that it's more suitable to our purpose. And that's what viral engineering has been used to do. And this is what we did in Berkeley during my second postdoc with John Flannery and David Schaper. And we applied this bioengineering technique called directed evolution in order to make AAV more suitable for retinal gene delivery and gene therapy. And what this engineering allows us to do is to make the virus more suitable to deliver genes into the retina, and also allow thereby to use less of the virus to obtain as efficient a delivery as we would need, which means that we don't get into trouble with the immune system because we're able to do what's called dose sparing. So use smaller dose, but get the same effect. And that's why viral engineering has been really useful to gene therapy.
Right. That's been such important work. And the work you did in Flannery's lab at UC Berkeley was really cutting edge at the time. So I congratulate you on that great work. And
thank you.
As you worked on making gene therapy more effective and safer, you started exploring some newer applications like optogenetics. And can you tell us how you first got interested in optogenetics and what you worked on? And I guess it helps for people to understand who may not know what optogenetics is. I know that's a lot of questions there.
Yes, so optogenetics was emerging just at the time when I started my first postdoc at Max Planck Institute of Biophysics, and Professor Bamberg that I worked with was actually one of the first people to have co-discovered and used it. So optogenetics is basically a combination of two technologies, optics for delivering light and genetics to deliver genetic material that makes a normal cell sensitive to light. So it's really like putting a solar panel onto a house. It allows to use this light energy for other purposes. It converts that light energy to control biological function within a cell in a nutshell. And how is this possible, you would ask, it's thanks to these opsins that have been found in prokaryotes, in microbes, so in algae and bacteria. So these are much simpler organisms than us, but they have our ancestral opsins. So these 7 membrane spanning proteins that are able to basically convert light energy into intracellular signaling we find equivalents in these ancestral microbes. And the advantage they have is that, unlike us, we use opsins through a complex phototransduction cascade to amplify the light signal and convert it into modulation of membrane potential. Whereas microbes, they do this in an all-in-one approach without any cascade. So it's much more simple. We have a direct conversion of light signal into membrane potential modulation, which means that we can really take the simple one protein and put it into any neuron of interest, and that neuron should automatically become sensitive to light without having to have any complex cascade behind it. This is the principle of optogenetics, which makes it so versatile and applicable, and which has made it so attractive to neuroscientific investigations, because up until the invention of optogenetics, neuroscience has been studying neural circuits and functioning of neurons via means like pharmacology or electrical stimulation that are much more coarse compared to optogenetics, which is making this available in a way that doesn't require any chemical or physical agent. So just by the use of light, you can manipulate circuits of neurons, break them, and understand how they connect and work together. So this is what has made a very big topic of interest from this field of optogenetics. And it's been used very, very widely for neuroscience applications. And the first therapeutic application in a biomedical aim has been suggested in the retina for restoring light sensitivity in retinas that have lost their endogenous photosensing capabilities due to loss of photoreceptors.
Right. And you were part of that, one of the first optogenetic therapies to move into humans with your work with GenSight. And in that application, if I understand correctly, the ganglion cells, which are sort of downstream from rods and cones, and they normally don't sense light, optogenetics was used to make them light sensitive.
That is right. This was indeed the first suggestion of use for optogenetics in the retina. It was actually during my first postdoc where we were doing more discovery of these different optogenetic proteins and their characterization that I read this article by Zhua Huapan's group. This was back in 2006, which was suggesting that this optogenetics could be used in ganglion cells for restoring vision, and that was what brought me to Berkeley, and then later to Paris Vision Institute, where Botan Roska and José Serral already had this vision of really bringing this from mice to patients. And I was very happy to be involved with GenSight Biologics, which is a spin-off of our institute with Botan Roska to participate in that effort of making this applicable in human beings.
Right. And along with GenSight, there are a few other companies, all of which have clinical trials for optogenetics. And in some cases, it's restoring some meaningful vision, which is exciting. But you mentioned a limitation of current optogenetics approaches, and that being you're using light-sensitive molecules from bacteria or algae to make the retina light sensitive again, and that has limitations. And if you could, I guess, reiterate those limitations and then talk about what you came up with to maybe do something even more powerful that's sort of like optogenetics, but not exactly.
That's right. So the limitation of optogenetics, although it's a fantastically robust and very compelling idea, is this origin of the proteins, of the opsins. So indeed, these are coming from prokaryotes, and obviously these bacterial and algae proteins are foreign to the human body. So that's why we had to work extremely carefully with GenSight to make sure that there is no adverse immune reactions. And thankfully, we're in the closed system of the eye where we have a privilege, and we enjoyed that immune privilege to adjust our doses and express just enough to get functional activation without without getting into trouble with these foreign bacterial proteins. So going further down, a lot of people actually imagine that maybe we could use our own opsins, but then there's this idea of the cascade. So how do you go around that? So I had already started thinking about these while the GenSight trial was ongoing. And the other problematic with the GenSight approach, which targets the ganglion cells, was that we targeted the retinal ganglion cells in an unselective manner. So normally, the ganglion cells are divided into many different subtypes, each having a very particular function in coding the visual scene and sending signals that are treating the signals and sending the signals from the photoreceptors to the brain. And that computational capabilities of the retina are actually so important to the way that our vision is so sophisticated with so many features. So when we go in there with a microbial opsin, irrespective of the ganglion cell type, and go and insert it in there, so all retinal ganglion cells then become light sensors. And we lose this information processing capabilities that they usually have. So we turn them into basically on and off switches. So that sort of degrades the quality of the restoration that we could hope, and is obviously an approach that is simple and robust, but less sophisticated than our natural vision, which is mediated by our cones, which come in 3 different colors, and that then signal to the bipolar and ganglion cells in very optimized ways as to form these very nice images that we see and we interpret with our brain. So messing with that and short-circuiting all of the circuit and going directly to the output with one type of channel is obviously bound to give a suboptimal result in terms of the restoration that we can expect, on top of the origin of this protein being also foreign to the human body. So those 2 points to many people were seen as points upon which we could improve in the next generation of products that we would develop in vision restoration. And one idea was actually Botan Raska and Jose's idea was to go directly to these so-called dormant cones. And there was a very nice science paper from 2010 where they had actually done exactly that. They had put a hyperpolarizing opsin into these so-called dormant cones, which are cones that lose their outer segment structures to a rod cone dystrophy and remain dormant because they're no longer able to perform the phototransduction cascade, which is found in their outer segments. So they were putting optogenetics, microbial opsins, directly into these cones and then benefiting from the information processing of the downstream retina. This was already a better way of leveraging what the retina knows how to do. But again, because it was microbial opsins, it was a single color and it was of microbial origin. So thinking about this, I actually ran into a German scientist called Stefan Berlitz. Stefan had a different idea of optogenetics. He was doing optogenetics using cone opsins in the brain by just fiddling with the C-terminal of the cone opsins and putting the C-terminal of the 5-HTA, the serotonin receptors, which are found in the brain circuits that are involved in depression and anxiety. He was able to actually use our cone opsins modified a little bit at the end with the serotonin receptor to modulate with light those circuits. So when I heard a neuroscientist using our cone opsins and being able to make them work in the brain, I thought, why couldn't we use these same opsins in the eye if we could kind of envision the same kind of operation? And what was happening was that in these serotonergic neurons that he was using, these cone opsins, there was this G protein activated inwardly rectifying potassium channel, which was acting as the relay from the cone opsin. So it was able to hyperpolarize those neurons in response to the G protein coupled signaling initiated by the cone opsin. So I thought, so why couldn't we do this in our cones? If we have these 2 elements, we could maybe recreate a short minimalistic phototransduction cascade that would allow us to bypass this problem of using opsins that are from our ancestral microbes. So that's the idea that gave birth to this SPVM20 concept. So I worked together with him on a grant and we started working around this concept in the retina. And what we did in my lab is we looked actually in mouse models at what happens to the phototransduction cascade in cones when they lose this outer segment? Is the whole thing gone, or are there some elements remaining? And actually, we were surprised to see that the cone opsin was still present in these dormant cones, but it just didn't have any more the downstream elements, especially the transducin and the phosphodiesterase, which are the downward steps from the opsin signaling, were very much downregulated. So it seems like the middle of the cascade was broken, but the beginning was present. So if we could give it a GERK channel with which to become operational again, we could do a sort of vision restoration with a minimalistic short circuit using only animal origin proteins. And that seemed like a compelling idea to me, also because using the endogenously remaining opsin gives us the possibility to envision color vision, because we have three types of opsins in our cones. When those cones go dormant, they still maintain those three types of opsins. So reawakening their remaining circuit via the GERK channel could also potentially lead to a restoration of color vision.
That's so exciting. And it really is ingenious that you looked at this brain researcher who was using retinal opsins or cone opsins and applying it back to these dormant cones. And I want to talk a little bit about dormant cones. And these are cones that have lost their outer segment, and that's sort of normally the light sensing portion of the cone, but the rest of the cone is still there. And you've done some studies, or other people have done some studies that show that a lot of people, even with advanced RP, still have dormant cones. Is that correct?
That's correct. Actually, I'm just coming back from Basel, where they had done a similar study for their optogenetic approach. This clinical study is called ICONIC, and they've looked at many RP patients and the maintenance of the foveal cones. We've done a similar study also at our Cansman Eye Hospital here with my colleague Isabel Odo. And Jose was actually one of the first people to point out to the possibility of these nicely remaining island of cones in the fovea of patients. So yes, today we have several lines of evidence that suggest that quite a few patients preserve these precious island of cones in the middle of their fovea, despite the loss of the outer segment structures that make them light sensitive.
Right. And could this even be for people who are very advanced with their RP, let's say, down to just light sensitivity? Do those folks have dormant cones in some cases?
It's really dependent on what kind of mutation you have and how advanced you are in many patient-specific factors. So there is several diseases where several types of mutations that are more prone to having this phenotype of dormant cones. But some of these patients could be quite far in terms of the natural history of the disease and still maintain them for decades on. So it's important, I think, that we really target those patients who are likely to benefit from this type of cone reactivation approach.
Certainly. And I think it's so exciting that you're reactivating the cells that are normally used for vision rather than going to ganglion cells or bipolar cells. You're using that, and you're using the natural light sensitive proteins or opsins. It's so elegant. And of course, we'll know better how well it works now that this trial is underway. And actually, before it moved into the trial, some time prior to that, you formed a little company, and that was acquired by SparingVision, correct?
That's correct. That company was called Gamma Therapeutics. That was back in 2020. And it all started out very small with this idea of using GERK. We also had a viral technology that went along with it. And within 1 year of the creation of the company, we started discussing with other companies for potential opportunities of working together. And SparingVision just turned out to be such a fantastic match because we had the same cell target and the same view of the importance of the cones. And with the approach that SparingVision was already working on, José Serral's broad-derived cone viability factor to preserve cones, it seemed like such an important synergy could be caught there. And so many common tools could, synergize both projects that we decided that this merger really made a lot of sense. And that's what initiated then the next stage of development for what became now SPVN-20.
Certainly, yes. Your approach is designed to restore function to dormant cones. The other emerging therapy from SparingVision, which has actually been in a trial for a couple of years, is designed to preserve functional cones. So you have yours for resurrection and the other approach, SPVN-06, for preserving. They're really great complementary approaches. And one thing I wanted to mention, because we're for the foundation, that we funded you for a lot of this lab research for this GERK approach that was ultimately acquired by SparingVision. And then we're also an investor through our venture philanthropy fund, the RD fund, in SparingVision. So we're excited to be supporting this work in multiple ways. But let's get back to the trial for SPVN-20. It just moved into a clinical trial. I think sites include Belgium, and then won't it be France and Ireland in Europe, if I recall?
That's correct, yes.
And then
the US, hopefully.
Right, next year. And so I asked this question reluctantly because we just won't know exactly what will happen until we see it in humans. But what do you think the potential vision restoration could be using this approach?
So the potential is really what I evoked earlier. So we're trying to make the most out of what's remaining and there. And I think that's the advantage we have compared to bringing something completely ectopic outside from, bacteria and bringing that in. So the advantage would be, using endogenous animal proteins, and GERK channels are expressed already in the brain, so they're not foreign to the human body. And there is this possibility of leveraging 3 types of opsins for trichromatic vision, color vision. So these are all the opportunities, but then there's also weaknesses and threats, as any project would have. So I think it's a lot less known in different patients who have these foveal cones remaining. What is the state of the cones and how long do they maintain these opsins? So we've looked, actually, thanks to an FFB initiative, there was a bank from RP patients who've deceased and who left their eyes to be examined for research purposes. We've looked at patients who are really elderly and who still had these dormant cones and were expressing opsin, but really our sample size was very small. So we don't know, broadening this to a larger population, what is going to be the amount of different types of cones and cone opsins within those cones that these patients will have, and bringing a certain dose of AAV to express these GERK channels. What is going to be then the stoichiometry between the remaining cone opsin and the GERK, and how is that going to work from one patient to the other, really at the molecular level, who have variations in terms of these expression patterns? These are the unknowns and potential difficulties that we could expect. So maybe what we have is something that is a little bit more fine in terms of the vision that we can bring back, but at the same time, it's less robust, because from one person to the other, there's going to be variations in its application. And I guess these are the things that we should keep in mind while, looking ahead with a lot of hope, obviously.
Right. And I think that's a really important point, especially for such an innovative approach that really hasn't been evaluated before. But when somebody is in a clinical trial, they're helping the research community learn, how the therapy works, who it will help. And clinical trials are still research. I know a lot of our constituents are always eager to learn about trials and potentially get involved, but we are still learning, especially in an early phase like that. Another question I have for you is what kind of test or imaging is used to determine if somebody has dormant cones?
And that's a really good question. So there's optical coherence tomography, which is a way of looking into the eye, non-invasive imaging method that allows us to see across the layers. This works quite well. And then there's also other techniques like adaptive optics, which have more cellular resolution, but they're a little bit more tricky because they're actually better at detecting the outer segments than the rest of the cones. So really, the studies that I referred to have used optical coherence tomography to look at the different retinal layers in patients and identify those with this foveal cones.
Got it. And that is known, the acronym is OCT. And a lot of researchers and just private practice retinal specialists have this technology.
That's correct.
That's cool. That's cool. So I don't want to get too ahead of ourselves because we're still learning about GERK, SPVN20, and we're still learning about SparingVision's earlier clinical trial for preserving cones, SPVN06. Hopefully, at least one or both will work. Do you think they could be used together or synergistically if we're fortunate and both work individually?
Absolutely. I think this is where we have the greatest potential to have a product that can be applied across a broader range of patients without asking ourselves too many questions about should we do this? Are we too late, too early? If we put everything together into those patients that have this phenotype and we're able to propose this one product that can, on one hand, keep the cones non-dormant for as long as possible. And if for some reason the cone does go dormant, then have also the backup plan to waken it back up all at the same time. This combined approach of 20 and 06, it's actually a product that that is in development at SparingVision called SPVN30. And we really hope that it would synergize both approaches and be able to help a much broader population of patients through the synergy of the two products.
That's so cool. I love that. Preserve the cones and resurrect the ones that are dormant to make them work again. That's very cool.
Exactly. Yeah, we can think of these dormant cones as, these cones that are not yet dormant when we intervene with SPVN6 with RDCVF. RDCVF is there to keep them awake so long as possible, keep boosting their metabolism, help them resist the oxidative stress that eventually makes them go dormant. But then if they do go dormant, GERK is there to wake them up.
I love that. I love that. Great concepts. And Deniz, you're still relatively young. I'm not going to ask you your age here on the podcast for the world to know, but you've come so far in your career with gene therapy development, work in optogenetics, and now GERK. I'm really impressed with your ability to innovate. So I'll put you on the spot a little bit. What's next? What are you working on now that GERK is in a clinical trial?
So that's a great question. So I think these concepts are so novel and so also fragile that we always, as we're developing them, are thinking about the next steps. So and through my career, I've been thinking about different parts of this problem. So there's the aspect of delivering the genes. So adeno-associated viral vectors and other types of delivery mechanisms are still constituting a core research activity in my lab. So we're still making the next gen vectors that are going to be more potent, less immunogenic, and are going to, in the end, help us solve the problems that are still associated with retinal gene therapy. So that's one activity. We're still working on the vectors. The other activity is we're still trying to come up with better strategies in terms of what to express in these remaining cells of the retina so that we can actually better reawaken them if needs be. So we're thinking not just about cones, but also how could we go into bipolar cells and ganglion cells? How could we diversify our toolbox? In this, at the same time, I think there's a lot of parallel developments in different types of technologies. So microbial options are getting a lot better. We have more sophisticated ideas for coupling GERK with other proteins to make them a little bit more, higher performing than what they currently are. So that's another axe of activity that is ongoing. And I'm also lucky to work with a professor of neuroimmunology in my group who's really looking carefully at these aspects of neuroinflammation around gene delivery and trying to find ways of dampening, alleviating, inhibiting, deviating those responses when they arise. So that we've been really looking at the basic science of this. So far we've been really lucky and benefiting from the immune privilege. But as you know, there's been some reports of adverse reactions to gene therapy in several clinical trials. So we're being really extra careful with these, especially knowing that we're coupling, we're not just putting back an existing protein like in gene replacement, but we're thinking of bringing exogenous things in. So that's another axe of activity in my group. And what broadly is really exciting to me is also I've been collaborating with a lot of researchers who are coming from a more theoretical background, because I've been finding our biology very lacking in numbers. So we've been trying to really think more numerically about a lot of these concepts of gene delivery and gene therapy and what is needed to restore visual activity. So I'm excited to put some numbers on our pharmacology with AAV, which is very different than pharmacology with small molecule drugs. So that's another axe, let's say, on which we're working and collaborating. So many different directions and many exciting things to come, hopefully in the next few years.
It sounds like great work, and it's exciting that you're taking the approaches that are just moving into humans and already trying to do better. You have a very innovative mindset. And on behalf of our listeners who may someday benefit from what you're doing, I just want to thank you for dedicating your career and your life to developing these technologies and continually improving them. And we're at such an exciting juncture, and we have you to thank, as well as many other investigators, for all your hard work.
And we clearly have, as this community, the foundation to thank for continuously supporting all of these different avenues, all in parallel, and really having confidence in our discoveries and in our ways of thinking, however improbable they might be, or however new or risky they might be. This continuous investment in our research is really helping all of this move towards patient application, which is obviously our ultimate goal.
Well, on behalf of the foundation, we're excited to fund such innovative work that has such great potential. So, Deniz, thank you again for taking time out of your busy day. You're calling in from France, I believe. And so you're toward the end of your day. I'm a little earlier in my day, but I appreciate you taking time to educate me and our community about all this great work. It's been so informative and frankly, very exciting. So thanks again.
Thank you, Ben. Thanks very much.
And I'd like to again thank Boehringer Ingelheim for sponsoring this episode, and to all our listeners, thanks as always for tuning in to Eye on the Cure. It's great to have you.
Thanks.
This has been the Eye on the Cure podcast. To make an impact, donate now at fightingblindness.org.